We are beginning Chapter 13 on meiosis and sexual reproduction. This video explores the processes that lead to the formation of gametes and the fusion of genetic material during sexual reproduction. Asexual reproduction involves cell division through mitosis. Mitosis produces genetically identical daughter cells, which are exact copies or clones. These cells have the same amount of DNA and the same number of chromosomes as the parent cell. Asexual reproduction occurs in single-celled eukaryotes like yeast, paramecium, and amoeba. Simple multicellular eukaryotes, such as Hydra, also reproduce asexually through budding. What are the advantages and disadvantages of asexual reproduction? Examples of asexual reproduction include budding in yeast and binary fission in amoeba. These processes create new individuals that are genetically identical to the single parent. What if a complex multicellular organism, like humans, wants to reproduce? Sexual reproduction involves the joining of an egg and a sperm. The crucial question is, do we make egg and sperm by mitosis? If we used mitosis, a 46-chromosome egg combined with a 46-chromosome sperm would result in a 92-chromosome zygote, which is incorrect. This image shows a human female karyotype, which is an organized profile of a person's chromosomes. It displays 23 pairs of homologous chromosomes. Similarly, this is a human male karyotype, also showing 23 pairs of chromosomes, including the distinct X and Y sex chromosomes. To maintain the correct chromosome number across generations, a special process is needed to make sperm and eggs. We must reduce the 46 chromosomes to 23. This is achieved through meiosis, which halves the chromosome number, followed by fertilization, which restores the 46 chromosomes. Meiosis is the process for producing gametes. Sexual reproduction involves alternating processes in stages. The chromosome number must be reduced from diploid 2n to haploid n through meiosis. Fertilization then restores the chromosome number from haploid n back to diploid 2n. Homologous chromosomes are paired chromosomes that carry genes controlling the same inherited characters. Both chromosomes of a pair carry the same information. In diploid cells, chromosomes exist as homologous pairs. These chromosomes duplicate to form double-stranded homologous chromosomes. Sexual reproduction involves fertilization. A haploid egg from the mother, containing one copy of each chromosome, fuses with a haploid sperm from the father, also containing one copy. This results in a diploid zygote with two copies of each chromosome. To make gametes for the next generation, a diploid cell with two copies of chromosomes must be divided to produce haploid cells with only one copy. This process involves mixing things up and reducing the chromosome number. Meiosis is a reduction division, a special cell division in sexually reproducing organisms. It reduces the chromosome number from 2n to 1n, or diploid to haploid, making gametes like sperm and eggs. Note, meiosis is radically different from mitosis and should not be confused with it. Meiosis is a double division. DNA replication occurs during interphase before meiosis begins. The first division of meiosis separates homologous pairs. The second division of meiosis separates sister chromatids, resulting in haploid cells with unreplicated chromosomes. The first step of meiosis involves duplication of DNA. This occurs because meiosis evolved after mitosis, and it is convenient to use the existing mitotic machinery. DNA is replicated during the S phase of interphase, just like in mitosis, resulting in double-stranded chromosomes for prophase I. The first division of meiosis separates homologous pairs. During prophase I, homologous chromosomes undergo synapsis, forming a tetrad. By telophase I, the cell divides, resulting in two cells, each with a haploid number of double-stranded chromosomes. Meiosis II is the second division, separating sister chromatids. Starting with the products of meiosis I, prophase II leads to metaphase II, where chromosomes line up. Anaphase II separates the sister chromatids, resulting in 4 haploid, single-stranded cells in telophase II. Meiosis I includes interphase, prophase I, metaphase I, anaphase I, and telophase I, separating homologous pairs. Meiosis II includes prophase II, metaphase II, anaphase II, and telophase II, separating sister chromatids, much like mitosis. Meiosis I begins with interphase, where chromosomes duplicate. Prophase I sees homologous chromosomes pair up and cross over. In metaphase I, tetrads line up at the metaphase plate. Anaphase I separates the homologous chromosomes. Meiosis II starts with telophase I and cytokinesis, forming two haploid cells. Prophase II proceeds to metaphase II, where chromosomes line up. Anaphase II separates sister chromatids, leading to 4 haploid daughter cells in telophase II and cytokinesis. These illustrations visualize meiosis I, showing the division that separates the homologous chromosomes, reducing the chromosome number from diploid to haploid. These illustrations show meiosis II, which separates sister chromatids. This second division is similar to mitosis, but starts with haploid cells and results in 4 haploid gametes. This chart compares the events of mitosis and meiosis, highlighting key differences in chromosome behavior and the resulting cells. Mitosis involves one division, produces 2 identical diploid cells for growth and repair, and has no crossing over. Meiosis involves 2 divisions, produces 4 different haploid gametes, and includes crossing over. Crossing over occurs during prophase I, when homologous pairs swap pieces of chromosomes. The sister chromatids intertwine, leading to genetic recombination. This process is essential for genetic variation. Crossing over involves 3 steps: crossover, breakage of DNA, and refusing of DNA, resulting in new combinations of traits. What are the advantages of sexual reproduction? Meiosis and crossing over introduce great genetic variation to a population, driving evolution. Random assortment in humans alone produces over 8 million different combinations. The value of meiosis is introducing genetic variation through recombination. The gametes of offspring do not have the same genes as the gametes from the parents, showcasing genetic recombination. Crossing over further increases variation by creating completely new combinations of traits in the next generation, as illustrated by the exchange of segments between homologous chromosomes. Finally, random fertilization dramatically amplifies genetic variation. Any 2 parents can produce a zygote with over 70 trillion unique diploid combinations, a product of the genetic mixing from both parents. Sources of genetic variability in sexual reproduction include independent assortment of homologous chromosomes in meiosis I, crossing over between homologous chromosomes in prophase I, and random fertilization, where a random ovum is fertilized by a random sperm. Sexual reproduction allows for maintaining both genetic similarity and differences. This can be observed in families, such as the Baldwin brothers, the Douglas family, and the Sheen family, who share traits while still being unique individuals. Differences in sexual reproduction exist across kingdoms. Not all organisms use haploid and diploid stages in the same way. For example, the dominant stage, either 2N or N, can differ, but they still alternate between haploid and diploid states for sexual reproduction. This concludes our video on meiosis and sexual reproduction. We have covered the process of meiosis and the sources of genetic variability it creates. Thank you for watching.